Hi everyone, this is Dr. Fry. Today we are going to be talking about angiosperm reproduction. Angiosperms, of course, are flowering land plants. They undergo a concept called alternation of generations, just like many of the other plants we've looked at so far. And just by way of a review, alternation of generations means that the multicellular haploid and diploid generations take turns producing each other. And the two generations are the sporophyte generation, which is our diploid generation, and the gametophyte generation, which is our haploid generation. So the sporophyte generation in angiosperms is what we would recognize as the mature adult plant. And if you remember, the gametophyte generation in angiosperms is going to be greatly reduced and in fact microscopic. So again, the sporophyte is a diploid generation. It will undergo some meiotic divisions to produce the spores, which of course then would be haploid after having undergone meiosis. And then there will be further mitotic divisions to produce the gametophytes. And the gametophytes would then still be haploid until they restore the diploid number when the gametes fuse. The gametophyte generation in the male for angiosperms is in the pollen grain and is called the microgametophyte. And for the female, it's the embryo sac, and that would be called the megagametophyte. And these will produce our gametes, so the sperm for the male and the egg for the female. And once those meet, just like in sexual reproduction in mammals, it will restore the diploid number, so the egg will be met by the sperm, restore our diploid number, and then these will continue to divide and grow into our mature adult sporophyte generation. Alright, so here is just that concept demonstrated in a nice graphic representation. So at the very top, you can obviously see that we have our flower. We have male and female reproductive parts within this flower. So the stamen is comprised of the anther and the filament. That's on the left hand side of our picture here. And then on the right hand side, you're looking within an ovule, which is of course in the ovary, which is the basal portion of the female reproductive part, the pistil. So the stigma is at the top of the pistil, the style is the shaft in the middle, and then the ovary is at the bottom. And of course inside there is the ovule. So let's just look at this picture to see a little bit more detail. First, I would like to point out one important detail, and that is that the color key in this figure is actually backwards. So it shows that the haploid generation is in blue up here at the top on both the left and the right sides, but of course that would be diploid until it undergoes meiosis, when it will become haploid. So the pink shading is actually the haploid, and the blue shading is actually the diploid generation. So those are backwards, so please make a note. I do think it's a very helpful figure though, so I'm including it. Just be careful with that one detail. So on the left hand side of our figure, you can see that there is a cut through the anther in this flower. And within there, it's showing you that there is a microsporocyte. And this microsporocyte undergoes meiosis to produce the haploid microspores. And I'll just mention that this prefix, micro, is often used to refer to the male reproductive parts. The counterpart on the right-hand side, you'll see that it says mega for a lot of the things. The mega prefix often refers to the female reproductive parts for angiosperms. So let's go back to the left. We talked about the microsporocyte undergoing meiosis to produce microspores. And there's a point before they divide where you can see that all 4 daughter cells from that meiosis are all sort of present within the same structure. And at that point, they're called tetrads. Once they divide, which they do, then they are the microspores. Okay, so as they continue to develop, they undergo some mitotic divisions and some differentiation, which will produce our male gametophyte. The male gametophyte is the pollen grain. It's within the pollen grain. And as you can see, if you, at the very bottom of the left-hand side, they're showing you a pollen grain, an actual picture of a pollen grain that we will look at also under our microscopes. And in that, there is a tube cell and a generative cell. The tube cell will produce a pollen tube, which will allow for sperm to eventually access the female reproductive parts and fertilize the egg. The other cell, the generative cell, will divide to produce two sperm. All right, so now let's just look briefly at the top right-hand picture. Before I move on, I want to point out that the top right-hand picture is a cross-section through an anther. And in this anther, you can see 4 pollen sacs. These are termed microsporangia. And inside each of these microsporangia is where all of that happens that we just discussed. The microsporocytes are the little dots within the microsporangia, and they undergo meiosis to produce the microspores, and then those will further develop and differentiate to produce our pollen grains, which will ultimately be released from the anther. All right, so now let's look at the right-hand side of our figure with the flower at the top. The right-hand side is our female reproductive development. So again, we are within the ovary. We are looking at one ovule within the ovary. And within that ovule, there is a megasporangium, which again is the counterpart to the microsporangium. So the megasporangium is where the megasporocyte will develop. The megasporocyte is the cell that will undergo meiosis to produce the surviving megaspore. Now, I should mention that the megasporocyte, it does undergo meiosis, but only one daughter cell of what would normally be 4 daughter cells for meiosis, only one daughter cell actually survives. So if you notice, then after the meiosis has happened, it says on the very right-hand side there, it says surviving megaspore. That's because the other daughter cells from meiosis will die off to conserve nutrients for the one surviving megaspore. That megaspore will undergo 3 successive mitotic divisions. However, the mitotic divisions will not be followed by a cytoplasmic division. In this situation, all of the nuclei from the 3 mitosis divisions will be present in one large cell. So the 3 mitotic divisions will result in 8 nuclei. These nuclei include 1 egg, 2 synergids, 2 polar nuclei, and 3 antipodal cells. And all of those can be seen in the picture at the very bottom right-hand side of this slide. So at the very bottom there, you can see there's an oval-shaped structure. This has 2 polar nuclei they are the most central of the nuclei, although they do tend to gravitate to the extremes. And then all the way on the right, you can see that there's an egg, and it is flanked by 2 synergids. And then all the way to the left, there are 3 antipodal cells, and those are each labeled there if you look closely. All right, so we're going to move on to the next slide. Microspore and sperm production. So this, as I mentioned, occurs in the microsporangium, which is within the anther. And of course, the anther is a part of the mature sporophyte generation. So the microsporocyte, as I had mentioned in our last figure, is a diploid cell which undergoes meiosis. So at the end of meiosis 1 and meiosis 2, there will be 4 separate cells, each of which are haploid, and these are then called the microspores. They undergo differentiation to produce our male microgametophyte, which will include the generative cell and the tube cell, all within the pollen grain. The generative cell further divides into 2 sperm, and the tube cell is a cell that produces a long tube called the pollen tube. And the 2 sperm will actually travel down that pollen tube in order to reach the egg and complete double fertilization. All right, so we are going to move on to our next slide. This is a picture of a cross section through an anther. And if you look at the bottom right, it says Lab Manual, page 98, slide 1. I have made some videos of each of the slides that you should have looked at if we had had lab this week. But obviously, you will not be doing that on your own. This is a picture, and the video that I've recorded for slide 1 are great representations, however, of what you would expect to see in slide 1, the lily anther first division, and what you are expected to draw in your lab manual on page 98. So the details that you should know for this slide are that this is a cross section through the anther. The 4 circles at each of the corners of the anther are the microsporangia, those circles. They hold the microsporocytes. The microsporocytes, again, are our diploid cell just before it undergoes meiosis to produce the microspores. The one little circle in the very center there, in the very middle, that is labeled the filament, and that is where the filament, which is the stalk, attaches to the anther. So there would be a lot of vasculature traveling through the filament to bring water and nutrients to and from the anther. So that is what you are responsible for the first slide. Again, I think it would be helpful to visit the video that I made for this slide as well, but just to hear some introduction to it first. Let's move to the next slide then. If we look closer within the microsporangia, if you see on the left-hand picture, we're within one microsporangia inside the anther. We've magnified that on the right-hand side. So now that you can see the division that is actually happening from the microsporocyte to form the microspores. And you can see there that it looks like where the pointer is pointed to, there are 4 distinct structures. These are the daughter cells from meiosis of the microsporocyte. So this is the highest magnification that we can get. And this would be very similar to what you would see for slide 2 that we expected you to do if you were having lab this week. So when there are 4, the daughter cells, those are called tetrads. Prior to that second division in meiosis, you would have had meiosis 1, which would produce what we call dyads. And that you can see just below where the tetrads are, just below where the pointer is, there's a cell that looks like it has undergone meiosis 1, and it looks like there are currently 2 daughter cells. That one will, of course, further divide to produce the tetrads, ultimately. All right, so let's move to our next slide. The release of pollen grains. The release of pollen grains has to happen through a break in the wall of the anther. And that is exactly what you are seeing here. So you can see that what had been two microsporangia has actually, the space has fused into one continuous area, and further, there is a break in the anther wall. The point at which the anther wall splits is called the line of dehiscence, and this split in the wall is what allows for pollen grains to be released out into our environment and go travel to another flower to fertilize the female reproductive parts. So this will be very similar to what you would be looking at on slide 3. It's a mature anther. It's undergoing a split in the anther wall. And if you look closely, you will see pollen grains have developed in the place of what had been our microsporocytes and our microspores. We will take a closer look at those on our next slide. So this next slide is showing you a pollen grain. At the very top is what it would look like under a microscope, and at the bottom right is a diagram to help you understand the different parts that should be in that pollen grain. So the pollen grain has two important cells, the tube cell and the generative cell. And you can see here that the tube cell is actually taking up most of the space in the top left picture. So within that pollen grain wall, there is on the left a nucleus of the tube cell. That is the nucleus, and the cytoplasm surrounds it. So all of that blue shading is the cytoplasm. And actually, the generative cell is within the cytoplasmic portion of the tube cell. The generative cell will further divide and give rise to two sperm, which will then travel down a tube, which is produced by the tube cell. So they are contained within the tube cell so that then they can travel down the tube produced by that cell. So in order to see that, let's move to our next slide. Here you can see that we've got what looks a lot like some pollen grains. The one in the very center has a tube extending from it, and the two dark purplish bodies in that tube, those are the two sperm, which have been created through a division of the generative cell. So this looks a lot like what we will see when we look at the video of slide 4, our pollen tubes. This should be very similar to what we're looking at here. All right, so that was all our male reproductive process, and this now is going to start talking about our female reproductive parts. The process is very similar between the two. As I mentioned earlier, anything with that micro prefix, that is going to refer to our male reproductive anatomy in the angiosperm. Anything with a mega, or sometimes even termed macro prefix, that will refer to the female angiosperm reproductive cells. So, as you can see, we're starting with our megasporocyte. So the mega, again, is going to refer to the female. The sporocyte is going to be what gives rise then to our megaspore. So the diploid megasporocyte will undergo meiosis. And as I had mentioned in an earlier slide, only one cell from what would normally be 4 daughter cells from meiosis, only one cell will survive as the successful megaspore to continue through the reproductive process. So as you can see, after meiosis 1, there's a big red X over one of those daughter cells. It does not continue to propagate. One of them does, but again, one of those 2 daughter cells dies off, leaving one haploid megaspore. And then that megaspore will go through 3 mitotic divisions. So the one cell will produce 2 nuclei. Then, of course, we will have 4 after the second mitotic event and 8 after the third mitotic event. So at the end of this, we have an 8 nucleate cell, which has not undergone any cytoplasmic division. So it's one large cell with 8 nuclei. One of the nuclei will be the egg. The other 7 will include 3 synergids, 2 polar nuclei, 3 antipodal cells, 2 polar nuclei, and 2 synergids. All right, so let's look at that development further in the next slide. All right, so here is at the very top left-hand side, this is a cross section through the ovary. And as you can see, within that ovary, there are 1, 2, 3, 4, 5, 6 ovules, so around in a circle within the center there. So each of those ovules can further be magnified and look inside the ovule, and what you would see at different points in development would look, at some point, it would look like the left-hand side of the picture on the bottom, and at some point further in development, it would look like the right-hand side picture. So these are the mitotic divisions of the megaspore. The megaspore was the one surviving cell after the megaspore site underwent meiosis. That megaspore undergoes 3 mitotic divisions, ultimately producing the 8 nucleate cell. But first it has to produce a 2 nucleate stage cell, then a 4 nucleate stage cell, and that's what you're looking at in the bottom left and right pictures. And now let's move on to our next slide to see our mature 8 nucleate female gametophyte. So this is after the third mitotic division, and you can see in greater detail all the way to the left in the bottom picture you have 3 antipodal cells. They're kind of stacked like pancakes almost all the way on the left hand side, and then 2 large polar nuclei. Like I said before, they're the most central of the nuclei. However, they do tend to gravitate towards one side and the other. And then all the way on the right, you can see that there's one egg, and on either side of the egg is a synergid. So there's 2 synergids. Now, I'll also mention, if you look at the top picture, the diagram is showing you that the egg is at the base of the ovule, and there's actually right in front of that, there's an opening. You can see that there's an opening in the walls of the ovule. The walls of the ovule, I'll mention also, there are layers of cells there that are called the integuments. And these layers of cells will ultimately become a seed coat. As this all develops, the ovule becomes a seed that can then be propagating the plant embryo and traveling somewhere else, or sustaining itself until the conditions are right for growth. So the seed coat will develop from the layers of cells called the integuments on either side of that ovule there. And the opening in the middle is called the micropyle. And if you look at the right top picture, you can see that there is a tube. This is from our tube cell. The tube will travel down and enter and at the micropyle and allow for the 2 sperm to enter. And one of those sperm will, of course, meet with our egg, which is one of our 8 nuclei in the mature female gametophyte. And the other sperm will meet with the 2 polar nuclei. The 2 polar nuclei are the 2 large, most central of our 8 nuclei. And so let's move to our next slide to evaluate that further. All right, so as I mentioned, there are 2 sperm. One fuses with the egg. This will produce our plant embryo, our diploid zygote, which will develop into the plant embryo, and that will further develop into our next generation of sporophytes. The other sperm will fuse with the two polar nuclei, and so this will result in a triploid nucleus. And this triploid cell, it will develop into the endosperm, which is a very starchy food supply for the growing embryo. It'll provide nutrients and starches and oils for that new plant embryo that will obviously have to develop from the seed. All right, so let's move to our next slide. All right, so we have already looked at the two right-hand arms of this life cycle diagram. This is, of course, a life cycle of an angiosperm. It starts at the very top with that flowering structure that is representing our mature sporophyte generation for angiosperms. And then the outermost loop, all the way on the right, is showing you the male reproductive parts developing, and that's what we just worked our way through. And then on the inner loop on the right-hand side, it's showing you the female reproductive parts developing. So those will ultimately join the two branches on the right of this life cycle. They will ultimately join at the point where fertilization occurs, because the outside loop, the male reproductive process, has resulted in two sperm. The inner loop has resulted in that mature female gametophyte, which includes an egg. And then when sperm and egg meet, of course, you get your diploid zygote. And then when the other sperm meets with the polar nuclei, you get your endosperm, and that is a triploid cell. And so we're now down at the very bottom left-hand side where it says fertilization or double fertilization. And at this point, what we have is a seed. The seed has resulted from now the embryo being formed. The diploid zygote will develop into a plant embryo, which ultimately will develop into our mature sporophyte generation again. But before it does, it spends some time packaged in a nice, neat little package called a seed. And that seed, of course, the seed coat comes from the integuments, which were those layers of cells on the outermost part of the ovule. And then the innermost part is the nutrient source, the endosperm, the embryonic axis, which has different parts, which we will look at on the next slide, and a few other anatomical terms that we should be familiar with. So as a seed, of course, this plant embryo can sustain itself for some time. It can also remain dormant for a period of time until the environmental conditions are appropriate for it to germinate. It can be carried some distance by wind, water, animals, etc., or it can be planted and germinate shortly thereafter. So it will spend some time as a seedling and then mature into the adult plant, which would start the life cycle all over again. So let's move to our next slide to look at a little bit more about our seed development. So I've said several times that the seed coat results from the integuments, and inside you've got your embryo, and you've also got the triploid cell, which will be our endosperm, our nutrient source. So if you look then, the embryo itself is sandwiched between either two cotyledons in a dicot plant or adjacent to a single cotyledon in monocots. These cotyledons sometimes take on the role of the endosperm, sometimes they do not. The embryonic axis itself is comprised of the radicle, the hypocotyl, and the epicotyl. The radicle is the embryonic root, so it will be the portion of the plant embryo that will grow into the soil, form the roots of the plant, and absorb minerals and water from the soil. The hypocotyl and the epicotyl are both portions of the shoot or stem. The difference between the two is the point where the cotyledons attach. Above that point, it is called the epicotyl. Below the point where the cotyledons attach, then we refer to that portion of the shoot as the hypocotyl, which makes sense if you think about the prefix hypo, below, that will be the portion of the shoot below the cotyledons attaching to it. All right, and so as we have all of this sandwiched in our seed. As it is developing, we also have development of the ovary. So the ovule will form our seed. The ovary, which surrounds it, will form our fruit in angiosperms. And the fruit has several roles, one of which is to make the seed transportable by being eaten by animals and carried some distance because they are attracted to the fruit. So let's move to our next slide now and just mention a few more details. So seeds, we mentioned, are formed in the ovule after double fertilization. The plant embryo will develop. It will form roots and shoots, and then it can enter dormant phase until the environmental conditions are proper for germination. And there are a few different types of seeds that we will look at. The first is a eudicot or dicot seed that has thick cotyledons. So I'd mentioned previously that sometimes the cotyledons can take on the role of the endosperm. In a seed with thick cotyledons, that is exactly what has happened. So the endosperm, we had mentioned, was going to be that nutrient-rich source of food, very starchy, for the growing embryo to sustain itself until it can start undergoing photosynthesis. So the cotyledons in a dicot seed with thick cotyledons have all that starch present. And then that's what you're seeing in the right-hand picture. There are two, because it's a dicotyledons, two cotyledons. You can see that the tan segments there are both cotyledons, and they can actually be divided readily, showing you that in the middle then, surrounded by the blue box, you can see your plant embryonic axis. And the plant embryonic axis has three different parts: the epicotyl, the hypocotyl, and the radicle. The epicotyl, again, is the part of the shoot above the attachment point for the cotyledons. The hypocotyl is the shoot which is below the attachment point for the cotyledons. And then the radicle will be the root. That's at the very bottom of the embryonic axis. And then if you also notice, there are little leaves coming off of the epicotyl. These are sometimes called foliage and sometimes called plumule leaves. And they will produce leaves for the plant once it starts growing and starting to undergo photosynthesis. All right, so let's move to our next slide where we see a dicot seed with thin cotyledons. And you still have two cotyledons. It's still a dicot, two cotyledons. However, in this situation, the endosperm is still present. That role has not been taken by the cotyledons. So you've got your two thin cotyledons surrounded by the endosperm. And then you have at the very bottom there, you've got your embryo, your plant embryo, including the hypocotyl, epicotyl, and radicle. Okay, so let's go to our next slide and look at a few more details. We have at the very top right here a picture of a bean seed, which is a dicot, and it has thick cotyledons. So I'll just point out a little bit of anatomy. On the very outside, the pinkish brown structure, that's the seed coat, which has developed from the integuments, and that can easily be removed. And when it's removed, you can expose the cotyledons. So if you look down below the red kidney bean, you can see a picture showing where the seed coat has already been removed. That's the very bottom. And then the cotyledons just above that, you can see easily split into two separate portions. And then you can even see on the very right hand portion, the right hand cotyledon there is the plant embryo. So the very tip of that is the radicle. It will lead to the hypocotyl, and then above the point where that cotyledon attaches to the embryo, we call that then the epicotyl. There is one other feature in the bean at the very top that I want to point out. That's called the oval hilum, and that is the point where the seed had been attached to the ovary wall. So that has left a little scar there called the oval hilum. And then if you look at the picture to the left on the top, you can see a diagram just showing you that the integument is what formed our seed coat. Then the megasporangium is what held our megaspore site, which divided then into our megaspore and led to the process of fertilization then. All right, so let's move to our next slide, showing two different types of germination that seedlings can undergo. So these two types of germination are epigeal germination and hypogeal germination. So epigeal germination is one type of germination which ultimately will produce a seedling from a seed. And in this particular form, the radicle will emerge from the seed first, and the hypocotyl, which again is the shoot below the attachment point of the cotyledons, that forms a hook that then ultimately pulls the cotyledons and the epicotyl and the plumular leaves. It pulls that above ground. So that hook will pull the rest of the plant, the seed, above ground, and ultimately it will straighten up, and then the shoot will continue to grow from this point. And there's a very helpful picture on the next slide. We will get to that. In this type of germination, however, I'll mention the cotyledons, they have a role in storage, so they've been storing nutrients for the embryo, and photosynthesis. So they are raised above the ground because they're pulled by the hypocotyl. The cotyledons will actually then undergo photosynthesis for a brief period of time. They will eventually die off, and the first foliage leaves will then take on the role of photosynthesis. The other type of germination is called hypogeal germination. So the radicle will still emerge first, and it's protected by a sheath called the coleorhiza. And then the shoot emerges second, and it's again protected by a sheath, but this one is called the coleoptile. So let's move to our next few slides so that we can see that visually. So this slide is showing you epigeal germination. Again, you start with your seed on the very left. The hypocotyl forms a hook which pulls the entire, the rest of that seed up through the ground, and it will ultimately straighten up. The cotyledons will help with photosynthesis for a brief period of time until then the foliage leaves can take on that role, that responsibility. And then the cotyledons will die off, fall off the plant, and then you would really only see the first foliage leaves on the base of that shoot there. But I'll also mention in this picture, you should be able to identify where the hypocotyl and epicotyl are based on where the cotyledons attach. So as you can see now that the plant has grown a little bit, it's a seedling, you can very clearly see and distinguish between the hypocotyl portion and the epicotyl portion of our shoot or stem. Okay, so let's move to our next slide. This is showing you hypogeal germination. This is in corn, which is a monocot. So you can see that there's actually two different structures which emerge, and the seed itself stays below ground. Hypogeal germination is when the cotyledons will stay below ground. So in this case, you can see the radicle emerges first. It is surrounded by a protective sheath called the coleorhiza, and then the shoot emerges second, it will obviously grow upward and the radicle will grow down into the soil. The shoot is surrounded by the coleoptile, which protects it as it emerges from the soil. And then from that will grow the rest of the shoot and the foliage leaves. So this wraps up our presentation on angiosperm reproduction and seeds and seedlings. If you have any questions, please feel free to reach out to me or your lab instructor, and we will be happy to help you. I thank you very much for listening and hope you're all doing well.